Hello everyone and welcome to the biology lecture series, and today we'll be discussing the NCERT exercise for the topic sexual reproduction in the flowering plants. So I hope you know all the concepts, okay, which are included in this chapter. Okay, so this will help you solve the exercise. Okay, so what are flowering plants? Flowering plants are the angiosperms. Angiosperms, okay, these are the flowering plants, means they flower. Okay, and you must be knowing about the structure of the flower. It consists of sepals, petals, androecium as well as the gynoecium. Clear? So let's start with this lecture. So what is the first question here? First question is, you have to name the parts of the angiosperm flower in which the development of the male and the female gametophyte takes place. Okay, so first of all, you should know what is the male gametophyte and what is the female gametophyte. Then only you can answer this question. The first one is the male gametophyte. What is the male gametophyte? The pollen grain is usually called the male gametophyte. Okay? And what is the female gametophyte? Female gametophyte is the embryo sac. Embryo sac is known as the female gametophyte. Okay? In angiosperms. So in the question it is asking where is the pollen grain forming and where is the embryo sac forming. Clear? So let's talk about this. So here male gametophyte, also known as the pollen grains. These are developed within the pollen sacs of the anther. Okay, so you know anther is a bilobe structure. It consists of two lobes. Okay, let's draw a rough diagram of the anther. Okay, it is bilobe usually. Anther means it consists of two lobes and it is dithecous, means each lobe contains two pollen sacs, and within the pollen sacs the pollen grains, they are formed. Clear? So these are the two lobes of the anther. Okay, this one is known as the pollen sac, and within the pollen sacs these are the pollens or the pollen grains. Clear? So pollen grains are known as the male gametophyte. So pollen grains are developed where? Within the pollen sacs of the anther. Okay, so what happens here? The pollen mother cell, okay, also known as the microspore mother cells, this undergo meiosis, okay, to form a tetrad. Okay, and then what happens? All these microspores then mature into the pollen grains. Clear? So what is happening here? The microspore mother cell, this undergoes meiosis to produce a microspore tetrad. A microspore tetrad is produced. Okay, and then this forms the pollen grain. Clear? So these microspore, these are haploid. Microspore mother cell is diploid. Pollens will also be haploid. Clear? So here what is happening? Meiosis. Meiosis gives rise to 4 cells. Clear? So this microspore tetrad, how is the microspore arranged? These are arranged tetrahedrally or okay, isobilaterally. Clear? So if this is the okay microspore tetrad, so it consists of 3 cells here. 3 cells will be visible, although 4 cells are present. So we are talking about the tetrahedral arrangement of the microspore in the tetrad. So there will be 3 cells that will be visible here. Okay, this is the second one. and you will have the third one. Clear? So this is the tetrahedral arrangement. You can draw it like this also. Clear? So this is a rough sketch that I have shown you. Then let's talk about the female gametophyte, which is the embryo sac. So female gametophyte in the angiosperm is produced within the ovules. Okay. So ovule consists, or within the ovule, embryo sac is present. Now, so female gametophyte known as embryo sac is produced within the ovule, which are contained within the ovary. So ovule is present within the ovary. Now, each ovule contains the female gametophyte, which is commonly referred to as the embryo sac. The embryo sac develops from the megaspore mother cells. So what happens? So this is the megaspore mother cell. Okay. This megaspore mother cell will be diploid. Now it undergoes meiosis to produce a megaspore tetrad. It produces the megaspore tetrad, which is linear. Okay. This is linear. It consists of 4 megaspores. Out of these, 3 will degenerate, which are present at the micropylar end. So megaspores which are present at the micropylar end, this will degenerate, and this one, this is known as the functional megaspore. This functional megaspore will undergo, okay, 3 mitotic divisions. So here mitosis, you can write. It will undergo successive mitotic divisions, and it will give rise to the embryo sac, which you know it is 7-celled and 8-nucleate. So the megaspore mother cell undergo meiosis, and the functional megaspore undergoes series of mitotic divisions. Eventually, the embryo sac matures to contain 7 cells, okay, including a egg cell, 2 synergids, 3 antipodals, and 1 central cell, which consists of 2 nuclei. So it is 7-celled and 8-nucleate. Clear? Now let's talk about the next question. The next question is differentiate between the microsporogenesis and the megasporogenesis. Okay, so in earlier question we were talking about the microsporogenesis and the megasporogenesis. Formation of microspore is the microsporogenesis, and formation of the megaspore is the megasporogenesis. Through microspore, what is produced? Pollen grains are produced, and through megasporogenesis, embryo sac is produced. Now you have to tell which type of cell division occurs during these events, and you have to name the structure formed at the end of these 2 events. Means you have to name the structure which is formed after microgenesis and after megasporogenesis. Okay, so this is clear, very straightforward. Here this question is, so let's talk about the difference between the microsporogenesis and the megasporogenesis. So microsporogenesis, it is the process in which a diploid microspore mother cells, so microspore mother cells, these are diploid, okay, means 2, and it contains 2 chromosome sets. This will undergo meiosis to form haploid microspore, which will be 4 in number. Clear? Whereas megasporogenesis, it is the process of formation of haploid megaspores from the diploid megaspore mother cells. Now microsporogenesis, it occurs inside the pollen sacs, which are present in the lobes of the anther. Clear? Whereas megasporogenesis, this occur inside the ovules. Here pollen sacs are also known as the microsporangia. Now pollens are produced by the microsporogenesis. So at the end of the microgenesis, pollen grains are produced. Embryo sacs are produced by the process known as megasporogenesis. We have already seen in the previous question. The megaspore mother cells divide meiotically to give rise to 4 megaspores that are arranged in a linear fashion. And that one of the functional megaspore, it undergoes what? Mitosis, and it gives rise to the embryo sac. Clear? Now, in microsporogenesis, arrangement of the microspore is tetrahedral, whereas arrangement of the megaspore is linear. This I've already told you. Okay, so mostly it is tetrahedral. I've shown you. If this is the tetrad, this is how the tetrahedral arrangement will look like. So only three will be visible here. Okay, so this is the tetrahedral arrangement of the microspores. Okay, so this is how microspores are arranged. Then if we talk about the megaspores, okay, so they are arranged linearly. 1, 2, 3, and 4. So this is the linear arrangement of the megaspores. Clear? Then all 4 microspores formed are functional. Okay, so 4 microspores which are formed by the meiosis, so they should be haploid. All of them mature to form the pollen grains. Whereas in case of the megasporogenesis, what happens? The 3 megaspores which are present at the micropylar end, this will degenerate. Okay, suppose these 3 degenerate which are present at the micropylar end. This will degenerate. And only 1 functional megaspore will be left. So this one is the functional megaspore, and this will undergo 3 mitotic divisions. Okay, and it will give rise to 7-celled and 8 nucleate embryo sac. Clear? Now let's move on. Now meiotic cell divisions are happening here. Okay, meiosis. Meiosis is known as the reductional division. So what is happening? In both megaspore mother cell as well as in the microspore mother cells, these both cells are diploid. So meiosis takes place in the diploid cells. Meiosis, it usually occurs in the diploid cells. So both megaspore mother cell and microspore mother cells, these are diploid structure, and they undergo meiosis to form 4 cells, 4 megaspores and 4 microspores. Now the structure formed at the end of these events are through microsporogenesis, pollen grains are formed, which is known as the male gametophyte, and through megasporogenesis, embryo sac is formed, which is known as the female gametophyte. Now next question, what is the next question? You have to arrange the following terms in correct developmental sequences. Okay, so here a sequence is given to you. You have to arrange, okay, these in correct developmental sequence. The pollen grains, sporogenous tissue, microspore is given to you, microspore tetrad is given, pollen mother cells, and male gametes. Okay, the sporogenous cell, these are diploid. These are the cells present in the anther. Then microspore tetrad, this is haploid. Pollen grains are also haploid. Pollen mother cell will be diploid and male gametes will be haploid. So you know meiosis takes place here during microsporogenesis. So these structure, sporogenous tissue and pollen mother cell, these should come first, clear, and then rest of the structure should come. So what should be the sequence here? Now following is the correct developmental sequence. First is the sporogenous tissue. So these cells, sporogenous cells, these are diploid. Okay, and some of the cells, okay, they become specialized and they are known as the pollen mother cells. So these will also be diploid. And this undergo meiosis then. Meiosis is known as the reductional division. So microspores which are formed in a tetrad, so these will be haploid. Clear? And then the microspore, each microspore will then mature into the pollen grains, which will again be haploid. And pollen grains, they further give rise to the male gamete, which will be again haploid. Clear? So what happens? This is the pollen grain. The largest cell is the vegetative cell. And this one is the generative cell. Now, generative cell, this will further form 2 male gametes. Clear? That's why we have written there, male gametes. Now, when the microsporangium is developing, every cell of the sporogenous tissue serves as a pollen mother cell. Clear? Giving rise to a microspore tetrad possessing 4 haploid microspores through the meiosis process. So this is known as the microsporogenesis. Now when the anther mature, these microspores dissociate and develop into the pollen grains. The pollen grains mature and give rise to the male gametes. Clear? So this is the correct sequence here. Now let's talk about the next question here. You have to draw a very neat and labeled diagram of a typical angiosperm ovule. Clear? So you have to draw the diagram of the ovule, how the ovules look like, and then you have to label it also. Different parts of the ovule. So you can see this is an image showing you the ovule, which is also known as the megasporangium. So microsporangium was the pollen sacs, and the ovule, this is known as the megasporangium. And within the ovule, embryo sac develops. Clear? We have already seen how the embryo sac develops. Clear? So here you can see all the parts are labeled here. Now this is an ovule, okay, which is also known as the female megasporangium, where the formation of the megaspore takes place, and megaspore will then form the embryo sac, which is known as the female gametophyte. This is clear? Now, these are the structures here. This is the chalazal pole or the chalazal end. Okay, then we have the micropylar end. Okay, micropyle forms where the integuments are lacking. Clear? So here no integuments are present. So this region is also the micropyle. This is the micropolar pole. Clear? Now, these are the integuments. So there are two integuments, outer integument and the inner integument. These integuments later form the seed coat, also known as the testa. Clear? These are the nucellus cells which are present in the ovule. This is the embryo sac which is developing within the ovule. Clear? So this is the micropylar end of the embryo sac. This is the chalazal end of the this is the chalazal end of the embryo sac. This is the micropylar end of the embryo sac. Clear? At the chalazal end, antipodals will be present. At the micropylar end, egg apparatus will be present, which consists of egg and the synergids. And in the center, you can see there is a central cell which consists of two polar nuclei. Here you can see funicle. Funicle is the stalk. Stalk of the ovule is known as funicle. And here you can see the hilum, which is also known as the scar on the seed. This is the scar on the seed, also known as the seed scar. Okay, so hilum is the point where the funicle is attached to the body of the ovule. So this point where the funicle is attached to the body of the ovule, this is known as the hilum. Clear? Now let's talk about the various parts of the ovule. So first is the funicle. The funicle, it is a stalk-like structure which represents the point of attachment of the ovule to the placenta of the ovary. Clear? So what does it represent? It represents the point of attachment of the ovule to the placenta of the ovary. What is hilum? Clear? Hilum is the point where the body of the ovule is attached to the funicle. I've already showed you in the image. Okay, point where the body of the ovule is attached to the stalk of the ovule, which is known as the funicle. Integuments, so they are the outer layers surrounding the ovule that provide protection to the developing embryo. And there were two integuments, outer integument and the inner integument. Now, micropyle. What is micropyle? So it is a narrow pore formed by the projection of the integuments. It marks the point where the pollen tube enters into the ovule at the time of fertilization. So you should remember this. The pollen tube, so you know when the pollen lands on the stigma, it will germinate and it will form the pollen tube. Now this pollen tube, it will enter into the ovule, okay, or the embryo sac through the micropylar end. Okay, so you should remember this. So it marks the point where the pollen tube enters into the ovule at the time of the fertilization. Then after entering into the embryo sac, it will release the contents, which are the two male gametes, into the synergids. Clear? Now next is the nucellus. So it is the mass of the parenchymatous tissue surrounded by the integuments. okay, from the outside. Nucellus provides nutrition to the developing embryo, and the embryo sac is located inside the nucellus. Clear? Now chalazal end. So there was one chalazal end. It is the basal swollen part of the nucellus from where the integuments originate. From the chalazal region, the integuments originate, and it is the basal swollen part of the nucellus. Clear? Now next question, what is meant by the monosporic development of the female gametophyte? Female gametophyte is what? It is the embryo sac. Now what is monosporic development? Okay, so development of the embryo sac can be monosporic, diasporic, okay, tetrasporic. Clear? So you know there were 4 megaspores that were formed, which are arranged in the linear fashion. 1, 2, and 3, and 4. This was the megaspore tetrad. Okay, so these cells are haploid because these are produced by the meiosis. Now, out of this, 3 will degenerate and only 1 will be left, which is known as the functional megaspore, and this will undergo mitotic divisions to form the embryo sac. To form the 7-celled and 8-nucleate embryo sac. Okay, so this is known as monosporic development. What is diasporic? Where 2 cells, okay, or 2 nuclei of the tetrad are used. What is tetrasporic? Where 4 cells are used. Now, monosporic development is most common in the angiosperms. Okay, so monosporic development is a development of the female gametophyte from one functional megaspore only. That's why mono. Mono means one. Okay, spore, from one spore only, one functional megaspore. Okay, now in angiosperms, a single diploid mother megaspore mother cells undergo meiotic divisions to form 4 megaspore haploid cells. Only 1 out of the 4 megaspores is functional, which forms the female gametophyte, while rest of the 3 megaspores, they will degenerate. Clear? So there was one megaspore mother cell which was diploid. This will undergo meiosis. So you know, after meiosis 4 cells are formed, because meiosis, it occurs in 2 phases, meiosis 1 and meiosis 2. So at the end of the meiosis, a tetrad is formed. Okay, this is the megaspore tetrad. And this will thus, okay, out of this 3 will degenerate. Okay, and that one is known as the functional megaspore. And this will give rise to the embryo sac by undergoing mitotic division. Clear? Now, next question. With a neat diagram, explain the 7-celled and the 8-nucleate nature of the female gametophyte. So they are asking what is the, okay, how can you explain the 7-celled and the 8-nucleate embryo sac which is formed in the angiosperms? So here is the solution. So this is the embryo sac of the angiosperms. Here is the micropylar end. Okay, here is the chalazal end. At chalazal end, 3 antipodals are present. So these cells will be haploid. At the micropylar end, egg apparatus is present, which is made up of egg cell and the 2 synergids. So these cells are also haploid because they are formed by meiosis. So this is known as the egg apparatus. Okay, then this one is the central cell, and central cell, it consists of the 2 polar nuclei which will be also be haploid. Clear? Synergids, they consist of the filiform apparatus. Okay, and filiform apparatus, okay, they guide the Pullum tube entry into the embryo sac. So we have seen in the earlier questions that the Pullum tube, it will enter through the micropylar end into the ovule, and then it will enter into the embryo sac. And filiform apparatus, these help in the, or it guides the Pullum tube entry into the embryo sac. So pollen tube will enter into the one of the synergids. Clear? And filiform apparatus helps, okay, in guiding the pollen tube entry into the embryo sac. Now, the female gametophyte is formed by the mitotic divisions, okay, of the functional megaspore, which is haploid. Now, the megaspore divide mitotically thrice. So there are 3 mitotic divisions that takes place in the functional megaspore. So functional megaspore, it undergoes 3 mitotic divisions to form 8 nucleate embryo sac, which is 7-celled. The process of formation of 7-celled 8 nucleate nature of the female gametophyte is mentioned below. Now the 2 nuclei are formed after cell undergoes the first mitotic division. So after the first mitotic division, 2 cells will be formed, obviously, because by mitosis 2 cells are formed. Okay, meiosis give rise to 4 cells, but mitosis give rise to 2 cells only. So from 1 cell, 2 cells are formed in case of mitosis. Whereas in meiosis, what happens? 1 cell first give rise to 2 cells. So these cells will be diploid, these cells will be haploid. Then these 2 cells will again undergo, okay, meiosis 2 and will form 2 cells. Clear? So this is what is happening in the meiosis. 4 cells are formed. These all cells are haploid. Clear? So the 2 nuclei are formed after first mitotic divisions. Then these 2 nuclei move towards the micropylar end first and one at the chalazal end respectively. Now these cells then they divide and redivide and form the 8 nucleate structure. Clear? So there are 3 mitotic divisions in total. Clear? Now consequently, there are 4 nuclei each on the either side. Now at the micropylar end, 3 out of the 4 nuclei differentiate into the egg cell as well as the synergids. So there is 1 egg cell and there are 2 synergids. Clear? At the chalazal end, what happens? 3 out of the 4 nuclei differentiate as the antipodals. And the remaining cells, each from either end, moves toward the center and they are known as the polar nuclei. These polar nuclei are also haploid. Now, therefore, on maturation, female gametophyte looks like 7-celled structure, which is 8 nucleate, because these 2 polar nuclei, these are present within the central cell. Clear? Now, next question. What is the next question here? What are chasmogamous flower? And can cross-pollination occur in the clastogamous flowers? You have to give reasons for your answer. Here, so chasmogamous flower, so these flowers are open, means the androecium and the gynoecium, okay, these are exposed to the external environment, whereas clastogamous flower, these are closed. Okay, so the stamens, you can say, as well as the stigma, this is not exposed to the external environment because the flowers are always closed. Clear? So in clastogamous flowers, this will promote the self-pollination. Because these flowers are closed. Clear? So this is also a kind of adaptation to produce the seeds. Okay, especially in the areas where pollinating agents are absent. So flowers, they remain closed and they undergo self-pollination. Clear? Whereas chasmogamous flowers, they are open. The anthers and the stigma, these are exposed to the external environment, so they will undergo the cross-pollination. Let's have a look at the answer. Chasmogamous flowers, these are the flowers that have open petals, means these flowers are open. Okay, such that the reproductive organs, which are the androecium and the gynoecium, or you can say the stamens as well as the carpel or the pistil, these are exposed to allow the cross-pollination. Clear? Now, chasmogamous flowers, these are small and they are closed flowers with unexposed reproductive organs. So these flowers are closed. Therefore, they cannot undergo the cross-pollination. However, they undergo self-pollination since the stigma and the anthers are present very near to each other. So these flowers, these are bisexual. They contain both the sex organs, both the reproductive structures within them. Okay, and they remain closed. That's why they undergo self-pollination instead of the cross-pollination. The next question, you have to mention the 2 strategies evolved to prevent the self-pollination in the flowers? Okay, what are the two strategies to prevent the self-pollination? So first one, there are two strategies involved in preventing the self-pollination in the flowers. First is the dichogamy. So what is dichogamy? Dichogamy is the reproductive strategy in the flowering plants. It involves the temporal separation of the male and the female reproductive functions. Okay, so there is temporal separation, means separation by time. So what happens here? In protandry, male reproductive organs, which are known as the stamens, these mature before the female organs, which are known as the pistil. This is known as protandry. And what is protogyny? Here the female reproductive organ, which is the pistil, they mature before the stamens. Clear? So because of this, self-pollination is prevented. So for the self-pollination, there should be a synchrony between the maturation of both the male as well as the female reproductive organs. But here what is happening? Stamens are developing, maturing first. Okay, so stamen will produce the pollens, but the stigma is not receptive. So in this case, what will happen? Stigma is not receptive because it has not matured. So it will not accept the pollens from the anthers. Here what is happening? In protogyny, stigma is receptive, but pollens are not produced because the stamens, these are not mature. So here, no pollens will be produced. Now, so this temporal separation prevents the self-fertilization within the same flower or the plant. Now, dichogamy promotes cross-pollination, obviously. So because of this, cross-pollination is promoted and it enhances the genetic diversity in the plant population. Now, next one, self-incompatibility. Okay, next one is a self-incompatibility. So what is happening here? It is a genetically controlled mechanisms in which the pollen grain of a flower are unable to grow completely on the stigma of the same flower. Clear? So next one is the self-incompatibility. Now let's talk about the next question here. Next question is related to self-incompatibility. So what is self-incompatibility and why does self-pollination not lead to the seed formation in self-compatible species? Clear? Let's talk about the answer now. So self-incompatibility, we have talked about it is the genetic mechanisms in the flowering plants that prevents the self-pollination and promote outcrossing. Outcrossing means cross-pollination. Okay, what does cross-pollination include? It involves the transfer of the pollen from one plant to the another plant. Now, in self-incompatible species, a plant's own pollen is unable to fertilize its ovules or produce viable seeds. Okay, and this is genetically controlled. This mechanism produces genetic diversity because it is promoting the cross-pollination. Okay, here the genetic, you can say different, you can say pollens, they have different genetic makeup and the pollens are transferred to the plant. Okay, some different plant which has some different genetic makeup here. So this will promote the genetic diversity within the plant population and it will reduce the likelihood of the inbreeding. So self-pollination will promote inbreeding and cross-pollination, it will reduce the inbreeding. Clear? And it will promote the genetic diversity. Now, let's talk about the mechanism of the self-incompatibility. Now, first one is the recognition. So self-incompatibility involves the recognition and rejection of the self-pollen by the pistil, which is the female reproductive organ of the same plant. Now, this recognition is mediated by the specific genetic factors such as the S alleles or the S genes, which control the compatibility interaction between the pollen as well as the pistil. Clear? So, the self-pollen are not recognized by the stigma. Clear? And this, okay, this prevents the self-fertilization and promote the cross-pollination. Clear? So there are some S genes or S alleles which are present. Clear? Now next one, pollen-pistil interaction. So pollen-pistil interaction is also very important here. This also determines whether the pollen will be compatible or pollen will be incompatible. Now, when pollen from the same plant, means self-pollen, attempts to land on the stigma of the pistil, it triggers a series of the biochemical reactions and signaling pathway within the pistil. Now, these reactions lead to the recognition and direction of the self-pollen, preventing its germination or growth within the pistil. So, pollen-pistil interaction is very much important here. Clear? So, there are some receptors present on the pistil that will recognize the chemicals that are released by the pollens. Clear? And whenever the pollen lands on the stigma, then it triggers a lot of biochemical reactions and signaling pathways, okay, that are involved in the recognition of the pollen or the rejection of the self-pollen. Now, another one is the pre-zygotic barrier. Okay, pre-zygotic barrier means before the formation of the zygote. Zygote, you know, it is formed by the fusion of the egg cell as well as the male gamete. Clear? So, pre-zygotic barrier, what is that? Self-incompatibility acts at the pre-zygotic barrier also. Okay, and it prevents the fusion of the male gamete. Okay, with the egg cell. Okay, so what is fertilization? It is the fusion of the male gametes. So male and the female gamete, they fuse together and they form a unicellular diploid structure which is known as the zygote. Clear? So here the fusion of the gametes is inhibited. This is known as pre-zygotic barrier. Clear? Now this ensures that the seeds are only produced through the cross-pollination, where pollen from the different plant fertilizes the ovules of the pistil. And this will also promote the genetic diversity. Now next one, let's talk about the reasons for the lack of the seed formation in the self-incompatible species. So why the seeds are not formed? First is the pollen rejection. So self-incompatible species have mechanisms in place to recognize and reject the self-pollen, preventing its successful germination and fertilization of the ovules. Now, as a result, self-pollen is unable to deliver the sperm is the male gamete, to the ovules, leading to the failure of the seed germination. So, pollen is rejected, it becomes incompatible. That leads to the failure of the seed formation because, you know, the ovules, they develop into the seeds after the fertilization. But here, no fertilization is happening. There is no fusion of the gametes. Clear? So, no fertilization, no seed formation. The next one is the genetic incompatibility. So, what is self-incompatibility? Is controlled by specific genetic factors within the plant. We have already talked about the S alleles or the S genes. Okay, so these are self-incompatibility genes. Now, if a plant possesses self-incompatibility alleles, they are incompatible with its own pollen, and self-pollination cannot result in successful fertilization and the seed formation. Clear? Now, next one, what are the reasons for lack of the seed formation? Again, so third one is the promoting of the outcrossing. So, because of this self-incompatibility, outcrossing is promoted. Outcrossing means cross-pollination. Okay? Now, self-incompatibility promotes the outcrossing by encouraging plant to receive pollen from the other individual. And this will lead to the increased genetic diversity. Clear? Because the pollen, this will have different genetic makeup, and the egg cell, this will have some different genetic makeup. So, this will lead to increased genetic diversity, and it will also lead to the decrease in the inbreeding depression, which is caused by the self-pollination and self-fertilization. Clear? Now, leading to increased genetic diversity and the fitness within the population. Now, outcrossing ensures that the seeds are produced from a diverse pool of the genetic material, enhancing the adaptive potential and survival of the species. This is clear? So, these are the advantages of the cross-pollination, you can also say. Clear? Now, we'll talk about the next question. Next question is based on the bagging technique. Okay, so let's read this question. So what is bagging technique and how it is helpful in the plant breeding program? Okay, let's have a look at the answer. Now, the bagging techniques help prevent the fertilization of the stigma by an undesired pollen. Okay, what is undesired pollen? The unwanted pollen. So what we want that, okay, during the artificial hybridization, okay, what we want to do, we want to create the hybrids that have the desired characteristic. So what we have to do, we have to make sure that the stigma is not fertilized by the undesired or the unwanted pollens. So these pollens, okay, these are present in the air, okay, and some pollens, they can be carried by the insects. So we have to make sure that the unwanted pollen does not land on the stigma, and only the desired pollen that we have selected, that only, okay, fertilizes the stigma. Okay, so this bagging technique is very useful in that. So what happens during bagging? So bagging techniques help prevent the fertilization of stigma by the undesired pollens that can be present in the wind, that can be carried by the wind or carried by the insects. Clear? Now the pollination by wind, this is known as anemophily. Pollination by insects is known as entomophily. So what you have to do, you have to cover the emasculated flowers. What are emasculated flowers? Okay, flowers in which the anthers are removed. Okay, so anthers, you know, these are the part of the male reproductive structure, and within the anthers, pollen grains are produced. So if you remove the anthers, pollen grains will also, okay, will be removed because pollens are produced in the anther. So by emasculating the flower, what you do, you can prevent the self-fertilization. Okay, so in case of the bisexual flowers, okay, what is happening? Both the female and the male reproductive structures are present in the same flower. So usually in what happens in the bisexual flowers, both male and the female, both male and female reproductive structures these are present in the same flower. Means both androecium and gynoecium are present in the same flower. Means both stamens and the pistil are present in the same flower. So there are high chances of self-pollination. Alright, so there are very high chances of self-pollination because both the reproductive structures, male and the female, these are present close to each other also. There are high chances of self-fertilization or self-pollination. So, but what we have to do, we have to, okay, we want that the stigma should be fertilized by the pollen that we have selected because it consists of all the desired characteristic, and we want these characteristics to be present in the hybrid. So what we do the emasculation, we remove the anthers using the forceps or the needle. Then what you do, you bag these emasculated flower with the poly bag or the butter paper. So this is known as the bagging technique, when you cover these flowers with the poly bag or a butter paper. Now, what is the benefit of the bagging technique? So bagging technique is beneficial in the plant breeding program during the artificial hybridization also. Now in this, only the desired pollens are made to fertilize with the stigma in order to produce the plants with desired characteristic. Clear? Now let's move on. Now how bagging technique works. So these are the steps that are involved in this bagging technique. So first is the emasculation. So what is happening during emasculation? Now before the flowers open, the male reproductive organs, which are the anthers, these are removed from the flowers to prevent the self-pollination. Okay, or to prevent the unintended or unwanted or undesired pollen, okay, to land on the stigma. So these unintended pollen can be brought or released by some other plants, some nearby plants. So this process is known as emasculation and is typically done manually using the forceps or using the scissors. Okay, so first step is emasculation. Then second one is the bagging then. Bagging with the polybag or with the butter paper. After the emasculation is done, the emasculated flower is covered with a small bag or a mesh cage to protect it from the pollination by external pollen sources. Okay, small bag or a mesh cage, or you can say using a butter paper also. You can do the bagging. You can use a poly bag also. So this bagging, this protect the pollination or fertilization by the undesired pollen, which can be brought by the wind or by the insects. So pollination by wind, this is known as anemophily. So wind is an abiotic, okay, pollinating agent. So pollination by wind is known as anemophily. Clear? So this is the abiotic pollinating agent. Then insects such as honeybees. Okay, pollination by insect is known as entomophily. So this is the biotic agent. Okay. Now, after bagging, the bag is securely tied around the flower to prevent any pollen from entering or leaving the flower. Clear? Now, how bagging technique works? Again, the third point, controlled pollination. So you can do controlled pollination. So once the emasculated flower is bagged, and you can also write here, and the stigma becomes receptive. So it is very important that the stigma should also become receptive. Then only the pollen will germinate. Okay, so after the emasculation and after the bagging, then you have to wait till the stigma becomes receptive. And then the pollen from the desired male parent plant is collected and it is carefully dusted or applied to the stigma of this emasculated flower. So this ensures the controlled pollination between the selected parents, and then the bag is resealed and to prevent any further pollination from the unwanted pollens. Now, next is the monitoring and the maintenance. So what happens? The bag flowers are monitored regularly to ensure the successful pollination and seed development. So after the pollination, what will happen? Fertilization will take place because the pollen, they carry the male gametes. Now these male gametes will fertilize the egg. So after the fertilization, the ovule develop into the seed and ovary develop into the fruit. Okay. Now after pollination, the bags are left in place until these seeds will mature, at which point they can be harvested for further evaluation and breeding purposes. Okay, this is clear. Now, what is the significance, okay, of this bagging technique in plant breeding programs? So first is we can do the controlled crosses. So bagging technique allows plant breeders to control the pollination and ensure the controlled crosses between specific parent plants. So what we do, we first select the desired parents. Okay, and if the flower is bisexual, we can then emasculate it. If the flower is unisexual, there is no need of emasculation because there are very less chances of the you can say pollination or self-fertilization. Clear? So this controlled crosses ensures controlled crosses. So this bagging technique ensures controlled crosses between the specific parent plants. Okay, these specific parent plant consist of this desired characteristic which we want in the progeny. Now this enables the breeding of the plant with specific combination of the traits and the development of new cultivars with improved characteristic. Okay, this is clear. Now second is the prevention of the unintended crosses. Now, by preventing unwanted pollination from external pollen sources, the bagging technique helps maintain the genetic purity of the breeding lines. This is very important. It can be used for maintaining the genetic purity of the breeding lines, and it also prevents the contamination from the undesired pollen. Okay, undesirable pollens which consist of undesirable traits. Now, this is particularly important for maintaining the integrity of the breeding populations and developing pure lines for further selection as well as the evaluation. Clear? Now, third one, efficient use of the genetic resources. The bagging technique facilitates the efficient use of the genetic resources by enabling the targeted transfer of the desired trait. So we want all these traits to occur in the hybrid, or in the progeny that we are producing. So that progeny will be of superior quality, that will be of called as superior variety. Some of the traits that we can select, so traits such as which will lead to the high yield. Some traits that can be select, some of these are like abiotic stress tolerance or biotic stress tolerance. Now, so these traits can be transferred from one parent to the another. It allows breeders to combine the best trait for the different parent plants and accelerate the development of new superior varieties with enhanced performance and adaptability. Enhanced performance means they have high yield. They will be high yielding. So there is a lot of food demand. Food demand is increasing day by day. So these superior varieties will have high yield. They will have high vigor also. High plant vigor. They can easily adapt to the changing environment. Because they have genetic variability. Clear? They can tolerate. We can also select the traits such as which will enhance the tolerance of this plant to abiotic as well as the biotic stress. Abiotic and the biotic stress. Clear? Now, fourth one, reduction of the genetic drift. So by controlling the pollination and minimizing the introduction of the foreign pollen or undesired pollen, the bagging techniques helps reduce the genetic drift and maintains the genetic stability of the breeding population over successive generations. Now, this ensures the reliability and consistency of the breeding outcomes and long-term success of the breeding programs. So consistency of the breeding outcomes should be maintained whenever we are breeding. So in the next generation, those characters should remain in the next generation also. It should be passed on to the next generation. So if there is a contamination or there is a pollination or fertilization by some other unwanted pollen, then what will happen? Those characters can be lost. So for the long-term success of the breeding programs, this bagging technique is very much important. Now let's talk about the next question. So next question is related with the triple fusion. So triple fusion, it occurs during the double fertilization. Now what is the question? What is triple fusion? Where and how does it take place? And name the nuclei involved in the triple fusion. So to answer this, you should first know about the double fertilization. So double fertilization takes place in the angiosperms, which are also known as the flowering plant. Clear? So what happens during double fertilization? The fertilization is taking place twice. What is fertilization? Fertilization is the fusion of two gametes. Clear? So first one, what happens? One of the male gamete it fuses with the egg cell. So this is known as syngamy. One of the male gamete fuses with the egg cell, so male gamete is haploid, egg cell is also haploid, and this will give rise to the diploid zygote, which is 2n. Zygote is a unicellular diploid structure. Okay. So you can write here, it is a unicellular diploid structure. Now what happens? There is one more fusion that is known as the triple fusion. So what happens during the triple fusion? The second male gamete, okay, which will again be haploid, this will fuse with the two polar nuclei. The two polar nuclei, they fuse together to form the secondary nuclei. Okay, how is this secondary nuclei formed? This is formed by the fusion of the two polar nuclei. Okay, so this will be diploid. Secondary nucleus will be diploid. Now, this results in the formation of a triploid nucleus known as the primary endosperm nucleus. So this will be triploid. Clear? So this is known as triple fusion and this is known as double fertilization because fertilization is happening 2 times. Now let's have a look at the answer here in detail. So triple fusion occurs when a male gamete fuses with the two polar nuclei within the embryo sac of the flowering plants. So embryo sac, you know, it consists of egg apparatus at the micropylar end. This egg apparatus consists of egg and the synergids. Then at the chalazal end, antipodals are present, 3 antipodals, and then in the central cell is present, which consists of the two polar nuclei. So these two polar nuclei are present within the central cell. Now the following event takes place in the triple fusion. The pollen grains get dusted on the stigma and germinate, giving rise to the pollen tubes that will enter into the ovule. Now the pollen tube passes into one of the synergids and releases two male gametes. Now one of the two male gametes fuses with the egg nucleus and forms the zygote, this is known as syngamy, and the other male gamete fuses with the two polar nuclei which is present in the central cell. And this forms a triploid endosperm nucleus. Now the nuclei involved in the triplet fusion are, you know now, it is a male gamete, one male gamete, which fuses with the two polar nuclei. That's why a triploid endosperm nucleus is formed. Clear? And the central cell, okay, it gets transformed into the primary endosperm cell, and primary endosperm cell is consists of the this endosperm nucleus, okay, which is triploid, and the primary endosperm cell then transforms into the endosperm, which is the triploid structure. Clear? The next question is, what do you think a zygote is? Why do you think that a zygote is dormant for some time in a fertilized ovule? Okay, so fertilized ovule will further develop into the seed. Clear? So within the ovule, embryo sac is present, and within the embryo sac, egg is present. Okay, so egg is fertilized by one of the male gamete and it forms the zygote. Clear? So why the zygote is dormant sometime? Now the dormancy of a zygote in a fertilized ovule serves several important purposes in the life cycle of the plant. Now here are some reasons why zygote may be dormant for some time after the fertilization. First is the developmental synchronization. Now dormancy helps to synchronize the development of the embryo with the favorable environmental conditions for germination and seedling establishment. Okay, so if the conditions are unfavorable, then the zygote can remain dormant. So in the dormancy state, what is happening? There is very reduced metabolic rate. Clear? Now, by delaying the germination until the conditions are optimum, plant increase the likelihood of survival and successive reproduction. So this is a kind of survival strategy, you can say. Okay, this is the kind of survival strategy that the plants have adopted. Clear? Next is the seed dispersal. So dormancy enhances the seed dispersal by ensuring that seeds are dispersed at the most advantageous time and the location for colonization and for the establishment. Clear? Now seeds may remain dormant until they are dispersed by the wind, water, animals or some other means, allowing them to reach the suitable habitats for the germination. Clear? So zygote dormancy also helps in the seed dispersal, and by remaining dormant, what happens? The seed remain dormant till they are dispersed by the pollinating agents such as wind, water and animals, and this allowed them to reach the suitable habitats for the germination. Clear? Now next is the seed bank formation. Now dormancy contributes to the formation of the seed banks. So in the seed bank, a lot of seeds are stored. Clear? So these seeds, they consist of the zygote which are in the dormant form. Clear? So this ensures the viability of these seeds also for longer time. Now dormancy contributes to the formation of the seed bank where the seed remain viable but dormant in the soil seed banks for extended periods. Clear? Now seed banks, they serve as a reservoir of the genetic diversity. So seed banks are very important. What is the importance? They are the reservoir of the genetic diversity. So all the seeds of lot of species, plant species, they are stored in the seed banks. Okay. Now, and this ensures, and the insurance against environmental fluctuations also. Now allowing plants to persist through periods of adversity and ensuring the population survival. So these seeds can be used, later on, to ensure the population survival. Clear? So this dormancy is so important, you can see here. Next is the avoidance of the unfavorable conditions. Now dormancy allows zygote to avoid the germination during the unfavorable conditions such as drought, extreme temperatures or low light levels. By remaining dormant, zygote can conserve the energy and resources until conditions improve and germination is more likely to succeed. So these are all survival strategies. Clear? So whenever there are favorable conditions, then the zygote can, again, it can start dividing and it will form the embryo. Clear? So you can see these are unfavorable conditions, drought, there is lack of water, and there is lot of sunlight, extreme temperatures, high temperatures or cold temperatures, and then low light levels. These are all unfavorable conditions, and whenever the favorable conditions, they appear, then the zygote will be again, it will be active. The next is the prevention of the precocious germination. Now dormancy prevents the precocious germination where the seeds germinate prematurely. What is precocious germination? When the seeds, they germinate prematurely before they have a chance to establish a root system. So root system is very important because through the roots, the minerals are absorbed. So this dormancy prevents the premature germination of the seeds. And before they have a chance to establish the root system or absorb sufficient nutrients from the soil. Now delayed germination ensures that seedlings have the best chance of the survival and growth once they emerge from the dormancy. This is also clear. Now next is the seed longevity. Dormancy contributes to the longevity of the seeds by protecting them from the physiological deterioration. So during dormancy, the metabolic rate is very low. So this ensures the seed longevity. So seeds will be viable for very long duration. So this dormancy protect the seed from the physiological deterioration. So there are a lot of metabolic processes that are going within the seeds. Clear? So this protect the seeds from physiological deterioration and damage during the storage. Now dormant seeds can remain viable for extended periods, allowing them to survive harsh environmental conditions and ensuring the persistence of the plant populations over time. So overall dormancy of the zygotes in the fertilized ovules represents an adaptive strategy. It is a survival strategy that enhances the plant survival, dispersal as well as persistent in the dynamic. Dynamic means ever-changing environment and unpredictable environment. Now, it is crucial aspect of the plant life cycle that contributes to the success and resilience of the plant populations in diverse habitats. Okay, next question, you have to differentiate between these. Epicotyle, hypocotyle, coleoptile, coleorhiza, integument and testa, and perisperm and pericarp. Let's quickly have a look at the differences. So epicotyle, it is the region of the embryo above the cotyledon. Hypocotyle, this is the region of the embryo below the cotyledon. Hypo means below, epi means above. Next is terminate at the plumule. And next is the hypocotyle, it terminates at the cotyledonary node. So what is cotyledonary node? So it is the junction between the epicotyle and the hypocotyle. Then epicotyle, it starts from the cotyledonary node, whereas hypocotyle, it starts from the radicle. Clear? Radicle is also known as embryonic root. Okay. Epicotyle is known as the embryonic shoot. Now, epicotyle, it develops into the upper part of the stem, whereas hypocotyle, it develops into that part of the stem that develops into the roots. Now, epicotyle, it elongates in the epigeal germination, whereas hypocotyle, it elongates in the hypogeal germination. Now, difference between coleoptile and coleorhiza. So, coleoptile, it is a protective sheath. Okay, coleorhiza, this is also a protective sheath and it is undifferentiated sheath. It is also a protective sheath. Coleoptile, what does it protect? It protects the young shoots in the cereals and the grasses. So, these are the monocots. Whereas coleorhiza, it protects the root tip of the germinating grass or the cereals. So, roots, they are underground. So, they can, whenever they are growing inside the soil, so there is a lot of pressure that are exerted against them by the soil particles. So, they can get damaged. So, to protect the roots, this coleorhiza is present. Now, this coleoptile, it comes out of the soil, whereas coleorhiza, it remains inside the soil. Coleoptile, it covers the plumule. So, plumule give rise to the shoot system. Okay, plumule will later give rise to the shoot system. So, you can say the coleoptile, it covers the shoot tip or the plumule, whereas coleorhiza, it covers the root cap and the radicle. Now, coleoptile, it breaks the seed coat and grows, whereas radicle, it breaks the seed coat and stops the growth. Next is difference between the integument and the testa. Now, integument, it covers the ovule. Okay, you must have seen during the structure of the ovule, there are two integuments, outer integument and the inner integument. Okay, and testa is the outer covering of the seed. So, integuments, these later on transform into the testa. Okay, testa is also known as the seed coat. So, it is the outer covering of the seed, testa, and integuments which are present in the ovule, these get transformed into the testa. Now, integument, the cells are living, whereas the testa, the cells are dead. Integument is the pre-fertilized structure. Okay, it is formed before the fertilization event is happening, whereas after fertilization, the integuments, they get transformed into the testa. So, testa is a post-fertilization structure, means after the fertilization. Now, in integuments, sclerids are absent. Okay, because sclerids are absent in the living cells, and in testa, sclerids are present. So, sclerids are also dead cells. Okay, that's why in testa, the cells are dead. Integument is one or two layers. Okay, and it is the protective covering of the ovule, whereas testa is one layered. Here, integument, it is the protective covering of the ovule. You have to remember this also. Now, the difference between the pericarp and the perisperm. So, here, pericarp, this is the outer covering of the seed or the fruit, whereas perisperm, it is a nutrient tissue surrounding the embryo. What is the location of the pericarp? It is found in the fruits. Perisperm, it is found in the seeds. What is the composition? It is composed of three layers, exocarp, mesocarp, and the endocarp. Whereas the perisperm, it is composed of the stored reserve food material. What is the function of the pericarp? It is the protection and dispersal of the seeds, whereas function of the perisperm is to provide nutrition to the developing embryo. Now, development of the pericarp develops from the ovary wall after the fertilization, whereas the perisperm develops from the nucellus of the ovule. Okay. Now, examples of the pericarp includes apple, peach, and tomato. So, pericarp, This is the covering of the fruits. So these are all fruits, apple, peach, and this tomato, which is a vegetable. Now, perisperm examples include coconut, corn, and wheat. So this is perisperm, which develops from the nucellus. This is found in the coconut, corn, and the wheat. Now, let's talk about the fruits. So next question is based on the fruits. Now, why is apple called as the false fruit? Okay, usually the fruit they develop from the ovary. So ovary, after the fertilization, they develop into the fruits, and ovules develop into the seeds. Now, why is apple called as false fruit, and which part of the flower forms the fruit? You have to answer this. Let's have a look at the answer. Now, apple is considered as a false fruit because the edible part that we commonly eat is not derived solely from the ovary. So only ovary usually develops into the fruits. But in apple, okay, the fruit doesn't develop from the ovary itself. Okay, it is not solely derived from the ovary of the flower. Okay, which is a defining characteristic of a true fruit. So what is true fruit? It develops from the ovary only. But in case of apple, the fleshy edible part, the part that we eat, is derived from the receptacle. What is receptacle? It is also known as the thalamus. So thalamus is the swollen part. Okay, it is the basal part. And on the thalamus, all the floral structures are attached, such as gynoecium, androecium, okay, sepals and the petals. Clear? So here the fruit is derived from the receptacles of the flower rather than the ovary. Now, what is receptacle? It is also known as the thalamus. This is the thickened part of the stem, okay, to which the floral organs are attached. Okay, this is the thickened part of the, you can say, stem. Okay, and all the floral organs are attached to it. What are the floral organs? Androecium, gynoecium means sepals and the these stamens, and the pistil as well as sepals and the petals. These are attached to the thalamus. Okay, now in the process of apple development, after fertilization occurs, the ovary begins to develop into the seeds here, while the receptacle swells and grows around the developing seeds. Clear? So receptacles forms the edible part here. Now this swollen receptacle becomes the main edible part of the fruit that we know as the apple. Now the ovary itself forms the core of the apple, housing the seeds within. So while the apple does contain seeds, which are the mature ovules, the bulk of the fruit structure and the part we typically eat is not derived directly from the ovary. Okay, that is why we call it as the false fruit. Clear? Now next question, what is meant by emasculation, and when and why does a plant breeder employ this technique? So what is emasculation? Removal of the anthers. So this is very much important during the, okay, artificial hybridization, where we have to, okay, create the desirable, okay, hybrids, which consist of all the desired characteristic. Now, emasculation is a technique used in plant breeding to prevent the self-pollination or self-fertilization, okay, or it also prevents the fertilization or pollination by the undesired or unwanted pollens. So this is done by removing the male reproductive organ known as the anthers from the flowers before they release the pollens. Now this process ensures that the flower will only receive the pollens from the desired male parent, which is known as the parent donor, that consists of all the desired characteristic. Okay, and this is known as controlled pollination, and this allows the plant breeder to control the genetic makeup of the offspring. Okay, so the offspring will contain all the desired characteristic, and this will be a superior, okay, variety. Okay. Now, plant breeders employ emasculation for several reasons. First is the hybridization. Now, when creating hybrid plants, breeder want to cross two different varieties or species to combine the desirable traits from the each plant. Now, emasculation ensures that the female parent, okay, it receives the pollen only from the chosen male parent, preventing the unwanted pollen from interfering with the desired crosses. This we have already seen. Okay, we do the emasculation and then we do the bagging. Okay, next is the elimination of the self-pollination. Though some plants have mechanism for, okay, self-pollination, which can limit the genetic diversity. So self-pollination, it will enhance the inbreeding. And okay, if there is lot of self-pollination, there is repeated self-pollination, then it can limit the genetic diversity. Clear? Self-pollination can lead to inbreeding depression. Okay. This will lead to the this will limit the genetic diversity. Clear? Whereas cross-pollination, this will promote the genetic diversity, genetic variation. Now, emasculation prevents the self-pollination by removing the anthers, promoting outcrossing and genetic variation. So, during outcrossing, there is a mix of the genetic material. There is a mixing of the genome of two different species. So, genetic makeup is different of the different species and they come together in the progeny, leading to the genetic variation. Next is the controlled pollination. So, by controlling which plants contribute to the pollen to the breeding process, breeders can selectively introduce specific traits into the offerings. Alright, now emasculation allows breeder to precisely manage the pollination and the resulting genetic combination. Let's talk about the next point, prevention of the unwanted crosses. Now, in some cases, breeder may want to prevent natural or accidental pollination by the other plants in the vicinity. Okay, so we don't want the pollination by the undesired pollens which are present in the vicinity. So this which could lead to the undesired crosses and compromise the breeding goals. So for long-term success of the breeding programs, we require that there should be no, pollination by the undesired pollens which are present in the air, or which can be carried by the insects also. So to ensure that the breeding program should be successful, this emasculation and bagging should be done. So emasculation ensures that pollination occurs only under the controlled conditions. Now coming to the next question. Next question is, if one can induce parthenocarpy. What is parthenocarpy? Development of the fruit without the fertilization. So this can be induced by the application of some hormones such as auxins. So if one can induce parthenocarpy through the application of growth substances, such as hormones, which fruits would you select to induce parthenocarpy and why? Now let's have a look at the answer here. What is parthenocarpy? Parthenocarpy is the development of the fruit without the fertilization. So usually the fruits are developed after the fertilization. So after fertilization, the ovary develops into the fruits and ovules develop into the seeds. So here no fertilization is happening. So the fruits which are produced, these are seedless. Seeds are produced without the seeds. And this can also be induced artificially through the application of growth substances such as auxins and gibberellins. So these are both plant hormones, these are phytohormones. Now when selecting fruits to induce the parthenocarpy, several factors come into the play, including the market demand, fruit quality, and practical considerations. Now, what are these practical considerations? You have to choose cultivars that are naturally parthenogenic, or they have been bred for parthenocarpy. Another practical consideration is that parthenocarpic fruit development can be sensitive to environmental factors such as temperature, humidity, and light. So you have to consider all these points before you select the fruits. Clear? Now, fruits devoid of the seeds such as watermelon and muskmelon, they are in great demand. Therefore, these varieties will be developed by the parthenocarpy. Now, next question is, you have to explain the role of tapetum in the formation of the pollen grain wall. So, tapetum is the innermost layer of the anther. Clear? This is the question here. Now, let's have a look at the answer. Now, the tapetum is a specialized layer of cells found within the anther of the flowering plants, which play a crucial role in the development and maturation of the pollen grains. The tapetum is located adjacent to the microspore mother cell or the pollen mother cells in the anther. Now, what is the role of tapetum in the pollen grain wall formation? First is the nutrient supply. So, tapetum, it supplies nutrition. It is a nutritive tissue providing essential nutrients, metabolites, and the precursor required for the development and maturation of the pollen grains. Now, these nutrients include carbohydrates, lipids, proteins, and sporopollenin precursors. So, sporopollenin, it is present in the exine of the pollen. So, exine consists of two layers, or pollens consist of two layers, exine as well as the intine. So, exine, it consists of a biopolymer which is very rough. Okay. So, this sporopollenin, this is present in the exine of the pollens. And the material for producing this sporopollenin, this is provided by this tapetum. Okay. Now, secretion of the sporopollenin precursors. Sporopollenin, it is a complex biopolymer that forms the major constituent of the pollen grain wall known as exine. Now, the tapetum secretes sporopollenin precursors such as fatty acid, phenolic compounds, and other organic molecules which are transported to the developing pollen grains. Now, third is the exine formation. Now, tapetum plays a vital role in the deposition and patterning of the sporopollenin, okay, which is present on the surface of the developing pollen grains. Now, sporopollenin precursors are polymerized and deposited on the surface of the pollen grain under the influence of the enzyme and metabolic processes which are happening within the tapetal cells. Now, next is exine sculpturing. The tapetum contributes to the sculpturing and the ornamentation of the pollen grain wall, which is the exine, by controlling the deposition and the distribution of the sporopollenin precursors. Now, this sculpturing pattern is characteristic of each plant species and can serve as a direct diagnostic feature for the taxonomic identification. So, this exine sculpturing, okay, which is done by this sporopollenin. So, there is a deposition of the sporopollenin in some unique pattern. So, this pattern can be used for the taxonomic identification of the species. Okay. So, this pattern is quite unique and this can be used as a characteristic or the diagnostic feature. Next is the protection as well as the support. Now, tapetum, it provides the physical support and protection to the developing pollens during their maturation. Now, it surrounds the microspore or the pollen mother cells, shielding them from mechanical damage and environmental stresses. So, it provides protection and support also. Next is the PCD, programmed cell death. It is known as the programmed cell death. Now, following its role in the pollen development, the tapetum undergoes programmed cell death, allowing for the release of the mature pollen grains. Okay, so release of the pollen grains are very important. Then only, okay, they will fertilize the egg. Okay, now this programmed cell death is very important for the release of the mature pollen grains. Then the degeneration of the tapetal cell releases the enzymes and other substances that facilitate the release of the mature pollen grains from the anther. Clear? So, it helps in the dehiscence of the anther, we can say. After the dehiscence of the anther, the pollens are released in the environment. So, in summary, tapetum plays a multifaceted role. It plays a lot of roles in the formation of the pollen grain wall, including the nutrient supply, sporopollenin synthesis, exine formation, sculpturing of the exine, protection of the pollen grains, as well as support of the pollen grains. Its functions are essential for the development, maturation, and release of the viable pollen grains, ultimately contributing to the successful sexual reproduction and plant fertility. Now, let's talk about next question. Next question is about apomixis. Now, what is apomixis and what is the importance of apomixis? Now, let's have a look at the answer. So, apomixis is the process of seed formation without the fertilization. So, here seeds are formed without fertilization. So, what is the difference between apomixis and parthenocarpy? Parthenocarpy is the development of fruit without fertilization, and apomixis is the development of seeds without fertilization. Usually, both the seeds and fruit, these are developed after the fertilization. So, these are the structures which are developed post-fertilization, means after fertilization. Clear? Now, what is fertilization? They are talking about the double fertilization here. So, what is happening during fertilization? The process of meiosis and the syngamy. Okay, to be precise, clear, syngamy is a part of the double fertilization. Apomixis inhibits the loss of the desired characteristic in a hybrid. Okay, so what happens during apomixis? There is no meiosis. So apomixis is a kind of, you can say, asexual reproduction. You can say it is a kind of asexual reproduction. Okay, so there is no formation of gametes. No gametes are produced. So gametes are produced by meiosis. So during meiosis, what happens? There is recombination. This recombination leads to the genetic variability. Okay. So what happens during recombination, or what happens during meiosis? There is recombination and there is independent assortment. So whenever there is independent assortment, this will lead to the segregation of the characters. So it might happen that the progeny, so in the progeny, there will be no desirable characteristic that we have selected. But during apomixis, what is happening? No gametes are formed. There is no meiosis. There is no independent assortment of the genes as well as the chromosomes. So desired characters are maintained in the apomixis. Clear? So you can write the desired characters are not lost or they are maintained in the hybrid. Now also the process of production of hybrid seeds through the cultivation is very expensive. Clear? So therefore apomixis is used for the production of the hybrid seeds also. Now let's have a look at the importance of the apomixis. First is the genetic stability. Clear? So this I've already explained you. Apomixis result in the production of the offerings that are genetically identical to the parent plant. So it is a kind of asexual reproduction. During asexual reproduction, only one parent is involved and the progeny, it is the clone. It is genetically identical to the parents. So the characters are maintained in the progeny, in the hybrid. Okay. But during the sexual reproduction, they can be lost because there is the segregation of the characters. Now, this genetic stability can be advantageous for the propagating desirable traits such as disease resistant, drought tolerant, or high yield. So these characters will be maintained in the hybrid. Okay, without the risk of genetic variation introduced by the sexual reproduction. Next is preservation of the hybrid vigor. So, in hybrid plants where two distinct lines with desirable traits are crossed, apomixis can prevent the genetic combination of the hybrid without the need of complex breeding programs or maintenance of the parental lines. Okay, so hybrid vigor is maintained. Next is the crop improvement. Apomixis can facilitate the propagation and distribution of the superior crop varieties by allowing for the clonal reproduction of the elite lines. Okay, so this can streamline the breeding process and accelerate the development of new cultivars with improved traits. So it can be used for the improvement of the crops by the clonal reproduction of the elite lines because the characters will be maintained in the progeny. Next is the reproduction in the polyploid plants. Okay, polyploid plants are those plants which contains more than two sets of the chromosomes. Apomixis is commonly observed in the polyploid plants where the complexities of the meiosis and gamete formation can be challenging. Now, it provides a mechanism for polyploid plants to reproduce efficiently and maintain their genetic integrity. Now, next point is conservation and restoration. In endangered or the rare plant species, what can happen? Apomixis can be valuable for conservation efforts by enabling the rapid propagation of the plants, okay, without the need of the pollinators or compatible mates. Clear? We don't require any compatible mate because sexual reproduction, it requires the compatible mates. All the pollens, they cannot germinate. The pollen should be compatible, then only it will germinate. And sometimes the pollinating agents are not present. Clear? So in this case, the plant species can be lost. It can become extinct. So we can use this apomixis method, okay, for the conservation of these species. So this can aid in the restoration of the population and habitats also. The next is horticultural application. Apomixis can be utilized in horticulture for clonal propagation of the ornamental plants also, fruit trees and other economically important species. It offers a reliable and the efficient method for the producing uniform plants with the desirable traits. So overall, apomixis offers several advantages in terms of the genetic stability, efficiency, practical applications in the agriculture, horticulture, as well as the conservation. Now, by bypassing the complexities of the sexual reproduction, apomixis provides a valuable alternative for the propagation and preservation of the plant species and improve varieties. Okay, so this is the end of the session. We have discussed all the questions which was related to this topic. Clear sexual reproduction in flowering plants. I hope you enjoyed this session. Thank you very much for listening.